establish chromosome rearrangements in Drosophila. And Bell will tell you when to go. There we go.
Okay. So today I'm going to be talking about chromosomal rearrangements. An example of a few are pictured here. But before I do that, I'm going to give a broad introduction into the evolution of genome structure before I get into some of the specific hypotheses that we're testing with regards to chromosome rearrangements. So between species, there can be differences in the total number of chromosomes. So for instance, some plant species can have chromosome numbers in the hundreds, whereas other organisms, such as the honeybee, might have far fewer. It's also possible that between species, the orientation and arrangement of individual chromosomes can differ. So despite our understanding and knowledge that the structure of the genome is fluid, we don't have a clear understanding of the evolutionary mechanisms that can lead to these large rearrangements. It's possible that the evolution of complex traits may be driving how genes are distributed and ordered along the genome, by controlling how often new combinations of alleles are created through the processes of independent assortment or homologous recombination. So recombination results from crossing over during meiosis, where its primary purpose is to ensure the proper segregation of chromosomes. However, it's also a mechanism for shuffling genetic variation and providing new combinations of alleles. So there's both theoretical and experimental evidence that suggests that natural selection is more effective and adaptation is more efficient in the presence of recombination. However, there's also this effect of recombination that it can break apart favorable associations. In the case of local adaptation, it can exchange maladaptive migrant alleles onto the chromosome. So there appears to be a battle between how often you can generate new combinations of alleles versus keeping those favorable associations together. A type of structural variation that can reduce or modify the rate of recombination are inversions. So inversions occur when there's double-stranded breaks in a chromosome, and the resulting fragment gets reinserted end-to-end in reverse orientation. Inversions have generated a number of fixed rearrangements between species, so for instance between humans and chimpanzees, indicated by the red lines here, and then also across the chromosomal arms of Drosophila. But besides existing as fixed differences between species, inversions can also be polymorphic within a species. And as I mentioned, inversions can reduce recombination, and in Drosophila, that's due to the formation of these inversion loops. So crossing over is reduced in these regions. There's evidence across a variety of different systems and species that arrangements are adaptive or maybe selected for in populations. In the specific case of the third chromosome of Drosophila pseudoobscura, which I'll be focusing the remainder of my talk on, there's evidence that it's adaptive because the frequencies of arrangements are clinally distributed along the American Southwest. And this is in the face of extensive migration. So moving from west to east, particular arrangement frequencies might increase or decrease. And this cline has been stable since its initial description nearly 80 years ago. So despite the ubiquity in nature, we don't have a clear understanding how inversions can become established or maintained within populations. There's 4 different classes of hypotheses that are used to explain how this process works. The first hypothesis is that inversions are established through their direct effects. So there's evidence that some inversion breakpoints disrupt the protein coding regions of genes or modify regulatory elements. There's evidence in several polymorphic human inversions that this is actually the case. The second hypothesis is that inversions hitchhike along with an adaptive allele. So this is where there's a single beneficial mutation sweeping through the population, and inversion happens to capture it, and then it gets brought along with it to high frequency. The third is through genetic drift. So there's been previous theoretical models that have predicted that the probability of fixation for an inversion increases in smaller effective population sizes. The fourth hypothesis is that inversions are established through the indirect effects of suppressed recombination. So we've shown previously that as expected on the third chromosome of pseudoobscura, in regions of the chromosome where multiple inversions overlap, there's a significant suppression of recombination. This would be beneficial for a number of non-mutually exclusive reasons. So for instance, the inversion could capture a region of the chromosome that's free of recessive deleterious mutations. It could be holding together multiple loci or multiple genes that interact beneficially, so a form of epistatic selection, or it could capture a locally adapted segment of the chromosome. So our work, we're taking an integrated approach by looking at both the genome and transcriptome of the third chromosome arrangement system in pseudoobscura. to test these different hypotheses and hopefully gain an insight into the evolutionary mechanisms that can establish inversions in natural populations. So we've sequenced the full genomes of 54 isochromosomal males that are representative of 6 different arrangement types. We've also studied gene expression in the transcriptome of those 6 same gene arrangements across 3 different life stages, so in larvae, females, and in males. And we also tested for differential expression in inversion heterokaryotypes. The third chromosome of Pseudo obscura, the arrangements are each named after the location where they were originally discovered. So for instance, CU for Cuernavaca or PP for Pikes Peak. And what this diagram is showing is the ordering of the syntenic regions for each arrangement. So with the exception of the hypothetical arrangement, all of these are still extant in nature. And because each of the inversions are single overlapping events, we can build this network here to infer the relationship among the different arrangements and determine which chromosome was derived from which ancestral arrangement. We have mapped the location of each of these inversion breakpoints using large insert Illumina mate pair sequencing. And to identify breakpoint regions, what we did is search the chromosome for regions such as pictured here. So this is where the reads are mapping further than their expected insert size and then reverse orientation. In no case did we find that there is a breakpoint that physically disrupted a protein coding region of a gene. We did find one case in larvae where the standard arrangement had greater expression levels than Pikes Peak in one boundary gene. But I think for the majority of cases, we see limited evidence that the inversions are maintained through their direct physical effects. For the nearly 2,700 genes on the third chromosome, we searched for signals of adaptive evolution to test the predictions of some of the other hypotheses. We developed a statistic which we call the population-specific branch length. It's very similar in nature to previously developed methods, but it's trying to search for genes that contain significantly high proportion of arrangement-specific allele frequency changes. So a strength of our data set that allows us to be able to calculate this statistic is that, as I mentioned, we can infer what the relationship is among the arrangements based on the ordering of those syntenic regions. And so for each gene, we can fit our data to this phylogeny and then calculate pairwise genetic distances and therefore estimate branch lengths. So what we can see is that in inverted regions of the chromosome, indicated by the 2 dashed vertical lines, we get an overall increase in the branch lengths. We also get a higher proportion of significantly long branch lengths in these regions. And this is the case for nearly every arrangement in our data set. So this seems to provide limited evidence for the notion that this is just a single adaptive mutation that the inversion happened to capture and get swept along with it through hitchhiking. So we see multiple genes within the inverted region showing evidence of adaptive evolution and contain arrangement-specific allele frequency changes. We also tested for the significance of association between sites showing evidence for adaptive evolution within arrangements. So we did this by estimating linkage disequilibrium, or LD. So this diagram is showing LD estimated across the entire third chromosome. So this is nearly 1.3 million segregating sites. What we can see here is that in regions where multiple inversions overlap, we get extensive LD generated. Within those regions, we see significant associations between sites across multiple loci that are showing evidence of adaptive evolution. So this supports some of the predictions of hypothesis number 4, that the inversions are maintained through the suppressed indirect effects of suppressed recombination. We also tested for differential gene expression among the arrangements. A major takeaway from this analysis is that when we performed an unsupervised hierarchical clustering, which is illustrated there at the top, across each of the 3 life stages, we get that generally recovers that cytogenetic phylogeny inferred from the orientation of these syntenic regions, so that the history of the arrangements is related to the pattern of gene expression. Among genes that are showing evidence of significant differential expression, we see that there's an enrichment in odorant and taste perception. So again, we see that there's multiple genes that have transcriptional differences maintained by the inversions. So this further provides limited evidence for that hypothesis number 2 regarding hitchhiking, and further supports that the inversions are maintained through the indirect effects of suppressed recombination. In addition to the PSBL, or population-specific branch length analysis, we also looked at signals of adaptive evolution, looking at high mean-derived allele frequencies, significant to GMAS-D results, that's that skewed site frequency spectrum, or unusually long haplotypes using a statistic called IHS. So within each arrangement, we came up with genes that are showing evidence of adaptive evolution if they tested significant for one of those tests, and also contained at least one arrangement-specific fixed amino acid difference, or showed evidence of differential gene expression. And what we find is that the proportion of genes showing evidence of adaptive evolution is directly related to the size of the inversion. So there's been previous theoretical models that predicted that if inversions are established with their indirect effects of suppressed recombination, that the number of loci involved in the local adaptation or epistasis would be directly related to the size of the inversion. We can see that relationship here in our data. We did not get a significant relationship in the uninverted proximal or distal regions of the chromosome. So among genes that are showing evidence for adaptive evolution, we also performed the gene ontology or GO analysis to look for common biological functions or protein domains that are enriched among those genes. We see again that there is an enrichment for sensory perception genes, so an odorant or pheromone binding. So this could support a model of local adaptation through the indirect effects of suppressed recombination. For the very last bit of the talk here, what I want to end with is a potentially intriguing result that we see in genes enriched in the limonene and pinene degradation pathway. So this could support a model of local adaptation or epistasis through the indirect effects of suppressed recombination. So limonene and pinene are chemicals or monoterpenes produced by a variety of different plant species. And in nature, they act as antifungals or as insect repellents. But what made this result particularly intriguing is that in the 1970s, there was work that showed that the composition of these two chemicals varied clinally along the American Southwest. So these are indicated in the red and green bars here. So you can see there's substantial variation in the same region that overlaps the species range of Pseudobscura and the clinal distribution of these arrangements. So although there would be much needed work to conclusively tie these links together, I think that this provides an intriguing insight. So despite the wealth of genetic knowledge that Pseudobscura has provided us over the last 80 or so years worth of research, we still know relatively little about its ecology or life history in nature. So this might provide an insight to being able to infer something about an organism's ecology or life history based on its genetics. And so with that, I would just like to thank my advisor, Steve Schaefer back at Penn State, and everyone else who made this possible, and my sources of funding. And I'd be happy to take any questions. Yeah.
So some of these clients also replicating in other continents.
Yeah.
So do you know anything about this final distribution of this limonene, whatever?
We have not examined that, and I don't think that the composition of those have been studied in other areas. Yeah, I don't know what the clinal distribution of those would be like in other areas. We just have the data for the American Southwest.